Hi guys, and welcome to our module on epithelial tissue. The epithelium or epithelial tissues are one of the four basic types of tissues in the human body. We have, aside the epithelial tissue, we have connective tissues, we have muscle tissue, and we have the nerve tissue. We'll talk about the other three in a separate module, but in this module, focus only on the epithelial tissue. So there are sheets, cells that tend to cover, they do three basic things. They tend to cover the body surface. That's one. They tend to form most of the body glands. So most of the glands present within the body are formed by epithelial tissues, and then they also tend to line the body cavity. So they line the outer part of the body surface, the inner part of the body cavities, as well as provide structural architecture for plants. So to explore a little bit more on what I discussed in the previous slide about the different places in the body we can find epithelial tissue, starting with the function of covering exposed surfaces or external surfaces. They tend to cover surfaces like skin, like the digestive duct, the GIT, the reproductive tract, the ureter, the bladder, the trachea, and the lungs. All these are exposed surfaces that are usually lined or covered by epithelial tissue. In terms of glands, they tend to give rise to a majority of glands within the body. So, sebaceous gland in the skin is made up of epithelial tissues. Mammary gland in the breast is made up of epithelial tissues. Salivary glands in the mouth, gastric glands in the stomach, intestinal glands, and then the mucosal glands elsewhere are all formed from cells that come together to form an epithelial tissue. And then finally, they tend to line internal passageways like blood vessels and the lymphatic system. So blood vessels have their internal lining being formed by the epithelial tissues alongside lymphatic cells that have also epithelial tissues forming them. And just a bit of other extra structures that I didn't mention in the slide. Some of the lining of the body cavities I described before, like the pleural space, the pericardial space, the peritoneum, all of these spaces are lined by epithelial tissue. So the peritoneum is made up of epithelial tissue, same with the pleural membrane and the pericardial membrane. Let's talk a little bit about the functions of epithelial tissues. There are four major functions of epithelial tissues. First is to provide physical protection, and that's by virtue of the fact that they tend to line different surfaces internally and externally. They also help in controlling cell permeability. I'll talk about one of the major features of epithelial tissues that makes this possible. They help in also providing sensations, and they also help in producing secretions through the glands in which they tend to form. Starting with physical protection. So, as said before, epithelial tissues tend to line surfaces. They line the surface of exposed structures that are in contact with the environment, either externally or internally. Some structures I mentioned before, like the skin, for example, is exposed externally, and as a result, it needs to be protected, and that protection comes by way of the epithelial tissue that lines it. The gastrointestinal tract, although it's an internal structure or surface, it is also in a way exposed to the environment. Whenever we are putting into our mouth, it's coming from the outside environment and goes into the GIT. So that exposure is also present there, and as a result, protection needs to be conferred on the gastrointestinal tract alongside other tracts, and this is done by way of the presence of epithelial tissues lining the surfaces. It also helps in preventing mechanical wear and tear, so it prevents abrasion. We'll talk about one of the very important features of the epithelial tissues in the sense of regeneration. So, due to the fact that it helps in mechanical wear and tear, epithelial tissue has this unique feature of being able to regenerate. So, even though we're having wear and tear continuously due to physical contact of surfaces, um, whether we're talking about internal structures like the GIT or external structures like the skin, um, the fact that they tend to regenerate helps in limiting that level of wear and tear, and helps or confers mechanical protection as shown here. They also confer biological and chemical protection against bacteria, viruses, and byproducts, and they help in limiting or reducing dehydration in the body. So the second function of the epithelial tissue is in the control of cell permeability. And the first thing is that to notice that the epithelial tissues tend to regulate entry and exit of substances to and from the body. And for them to also do this, they also need to be sensitive to a number of stimulus that either increase or decrease entry of substances. Examples, for example, we have the small intestine lined by epithelial cells. So the epithelial tissue in the small intestine usually tends to be sensitive to parathyroid hormone or vitamin D, and as a result, when there's an increase in the amount of parathyroid hormone or increase in the amount of vitamin D, the epithelial tissues on the small intestine will lead to or will respond by increasing calcium absorption. Another example would be that of the absorption of iron when there's an increased amount of vitamin C present in the small intestine. So small intestine is also sensitive to the presence of vitamin C or ascorbic acid in the GIT. And once that is present, it also tends to promote iron absorption. So these are ways in which epithelial tissues across different aspects of the body could be involved in regulating cell permeability. One other example is that of the epithelium of the loop of Henle. In the loop of Henle is lined by a number of different cells, that are epithelial cells, and these cells are also sensitive to various substances of stimuli. For example, aldosterone, which tends to trigger the increase in absorption of sodium across that epithelial tissue. So these are just different portions of different sites in the body lined by epithelial tissues that help in regulating entry and exit of different substances to and from the body, and due to the fact that they are very sensitive to various stimulus. The third function of the epithelial tissue is in the provision of sensation. Now, this is a very unique feature of or unique function of the epithelial tissues, and it's only seen in special sense organs. So, organs that are involved in special senses, like the eyes, the ears, nose, are also made up of specialized epithelial cells. This form of epithelial cells are known as neuroepithelium, and their major function is in the provision of sensation, and they tend to have large innervations by sensory nerves, and they tend to contain a number of different receptors. They contain receptors for vision, receptors for audition, for olfaction, and even on the skin, even non-specialized epithelial lining like the skin also has large amount of receptors that are also there to provide sensation. So on one end, we have specialized epithelial cells found in those special organs, special senses organs that are involved in special senses like vision, audition, and olfaction. And on the other end, for a typical organ that is lined by an epithelial tissue like the skin, it also is enriched with a lot of nerves and a lot of receptors that are involved in sensing different stimulus. So some of these receptors found in the skin would be things like mechanoreceptors, there's electrical receptors, and there's chemoreceptors found within the skin that also helps in sensing stimulus and providing sensation. The final function of the epithelial tissue is in the production of glandular secretion. There are two major forms of glands. We'll talk about that later in the slide, but we have the exocrine gland secretion and the endocrine gland secretion. We'll talk about the difference between each of them very soon. In terms of the exocrine gland secretion, this includes things like sweat, tears, milk. So we have different glands, exocrine glands formed by epithelial cells that are involved in the secretion of each of these substances. So they usually tend to release these substances into special sites we call ducts, and from there, ducts are eventually going to drain the secretion out to the desired site. And we have endocrine glands. Endocrine glands are also involved in secreting particular substances, but these substances tend to be released into the blood or lymph, and they tend to have action elsewhere. The particular form of substances that are produced by endocrine glands are usually termed as hormones. So hormones are produced from endocrine glands, whereas we have various forms of secretions like sweat, tears, and milk produced by exocrine glands. Now let's talk in particular about features. Features that tend to make a particular type of tissue to be classified or characterized as an epithelial tissue tends to be five in number. So there are five things you should see in a tissue that will make it to be characterized as an epithelial tissue. First is the fact that these tissues contain cells that are held tightly together by what we call cell junctions. Second is that each of these cells tend to have or exhibit polarity. Third is that each of the cells tend to have an underlining, or each layer of the cells tend to have an underlining basement membrane that tends to anchor the cell tightly to to a connective tissue. The fourth is that the cells are avascular. They tend to lack blood vessels within them. And then finally, the epithelial cells tend to have high rate of regeneration, tend to regenerate, especially through the fact that they have stem cells. There's some other slight features that we might not see in all epithelial cells, but are unique to a few of them. So, for example, presence of keratin found in epithelial cells of the skin is one. Presence of microvilli found in epithelial cells like that of the small intestine is another. Presence of cilia epithelial cells are found in epithelial cells that are present in those special organ sites I mentioned, like the ones in the ear, the one in the eye, the one in the nose, and even in epithelial cells found within the special sites like the testes and the ovaries. Let's start with the first major feature of the epithelial cells, the fact that they tend to be made up of cells, group of cells that are held tightly together by intercellular junction. So these intercellular junctions are specialized macromolecules that are responsible for holding epithelial cells tightly together. The cell-to-cell junction that is formed between each cell tend to have three major roles. First is to maintain integrity, and this is the reason why epithelial cells are very unique in the ability to to provide protection, because the cells themselves are tightly held together, so they don't allow for easy break in a particular surface that they are lying. So think about the skin, for example. If the cells that are forming the epithelial lining of the skin are not held together tightly, just by touching it, you could have of a breakage of the texture or integrity of the skin as a result. And we do see this in a number of conditions. So conditions like bullous pemphigoid or pemphigoid um, um, impetigo tend to have issues with presence of um, some of these intercellular junction, and as a result, you could have issues with an integrity of the skin from there. So I wish I had pictures to show you guys, but if you could Google, you check pictures of bullous impetigo or pemphigoid vulgaris or pemphigoid impetigo, you would see different pictures of the epithelial lining of the skin just falling off as a result of that lack of integrity due to the fact that the cell junction is missing. Aside from maintaining integrity, the intercellular junction also tend to serve as a form of support, and they also tend to have they confer communication between each of the cells. So one cell can communicate with another cell by virtue of specialized forms of intercellular junctions. For example, the gap junctions are very important in terms of communication between cells. So some of these intercellular junctions I mentioned before, just to touch on them a bit, we have three major intercellular junctions for you to know about. We have the tight junctions, the gap junctions, and the desmosomes. They tend to join one cell to the other, and they hold them they hold them tightly together and tend to confer those three major features or functions I mentioned. That's maintaining integrity, support, and communication. To talk about each of them one by one, tight junctions, for example, tight junctions are found between two cell membranes. This is a picture of a tight junction. You can see this is one cell membrane here. This is other cell membrane here. You can see how they're held tightly by tight junction. What tight junctions tend to do is that they form an adhesion belt, uh, and they tend to attach to terminal webs, and they prevent passage of water and solute or acid and bacteria between cells as a result, because they tend to limit passage of structures between the space between the cells. So they, in doing so, isolate waste, metabolic byproduct, and different structures in the lumen, and prevent the cells from or prevent the structures from passing through or between the cells to come out of a particular organ. For gap junctions, like I said before, are unique in the fact that they tend to create that space of communication between two cells. So they serve as point of communication between cell one and cell two. They are found in very unique structures like the cardiac cells, cardiac tissue, and even some other epithelial tissues. Then we have the desmosomes and the desmosomes. So let's talk about the desmosomes. Desmosomes include structures like intercellular cement, the camp structures, and dense areas. That what they tend to do is that they tend to tie cells together. So they do virtually the same thing that tight junctions do. They tie cells together and they're usually supported internally by an intermediate filament. Usually, what tends to anchor these desmosomes to one another, or anchor it very well, is usually the intermediate filaments found within the cell. So you can see one is cell 1, this cell 2, and we can see the intermediate filament that tends to anchor it. That's the desmosomes here connecting the cells together. This is in contrast to the tight junctions. In tight junctions, we have addition belt. The addition belt is what is connecting them together, and we have a terminal web inside the cell. So these are the terminal webs inside of the cell. So the terminal webs for the tight junction is similar to the intermediate filaments that we see in the desmosomes. Desmosomes are unique in the sense that they also tend um, to allow for bending and twisting of the cell. So, unlike the tight junctions that just create that tightness between cells, desmosomes allows for a bit of flexibility and allow the cells to be able to bend and twist alongside their being held together with and maintaining the integrity of the tissue. We have in another form of intercellular junction. However, this is not necessarily an intercellular junction. I'll talk about it when talking about the basement membrane, and that's called the hemidesmosome. So hemidesmosome is a unique cellular junction that helps to anchor a cell to the basement membrane. So this is not an intercellular junction because it is not anchoring the cell to cell; it's anchoring the cell to the underlying basement membrane. I'll talk about that when we get to the function or feature of the basement membrane. The second unique feature of an epithelial cell or epithelial tissue is the presence of polarity in each of the cells. Polarity just means that there are two ends that are distinct in a cell. So there's a basal pole or basal end, and there's the apical pole or apical end. So think about the basal pole as the floor of the cell. So if you have layers of epithelial cells as I'm drawing here, so let's say we have three layers of epithelial cell here. This is cell 1, this is cell 2, this is 3. What I mean by polarity is the fact that each of these different um, top and bottom of the cell are different poles or ends. So this would be the basal pole, the part that is sitting down, or what I call the floor, and this would be the apical pole, the part that is pointing up, what I call the roof. The apical pole tends to face the lumen. So if we have one of the an epithelial tissue lining a surface that is internal, like a GIT or the respiratory tract. Each of these tracts they have lumens within them. So the part of the epithelial tissue facing the lumen is the apical pole. The part of the epithelial tissue that is sitting on the basement membrane. Remember that every single epithelial tissue has a basement membrane where they are anchored to. So the part that faces the basement membrane is the basal pole. So the basal pole is the part of the epithelial tissue that is tightly held to the basement membrane. Remember that I talked about hemidesmosomes as the anchor points between the epithelial cells and the basement membrane. The part of the epithelial tissue that is facing the lumen is the apical pole. And the apical pole could be modified for that to have some of those features I mentioned before. Remember I mentioned there's some unique features that we don't see in all epithelial tissues, we can see in some. So some features like microvilli found within the GIT intestine, for example, is a picture of microvilli here, would be found on the apical pole of epithelial tissue. Or the cilia dimension, cilia that is found in a structure, some of those special sense epithelium or within the ears or within sense organs, they usually contain cilia, and the cilia tend to be found also on the apical pole of those epithelial tissues. And then the flagellum as well. Each of these different modifications have their roles. So, for example, the microvilli present in the intestine helps in increasing the surface area of the epithelial tissue. They are like finger-like projections of the plasma membrane on the apical surface, and they are involved in increasing the surface area either for absorption or for secretion of substances. The cilia that I also mentioned is there to provide a form of whip-like motile extension of the apical surface. They tend to lead to movements. They help in allowing movement of substances around where that particular epithelial tissue is found. And then the flagellum are usually long, very long isolated cilium. So they are cilia, but they are longer than in the normal cilia, and they are found usually in sperm cells. Sperm cells are very unique cells that um, tend to originate from structures found within the within the testis, and the entire reproductive tract themselves has the reproductive tract of the male and even reproductive tract of the female, like uterine duct, they have structures that are ciliated and structures that all a structure like the sperm cell would have a flagellum at the end of which is an extension of its apical surface. The third feature of the epithelial tissue is the presence of a basement membrane. Basement membrane tends to serve as an anchor for the epithelial tissue. It's like the foundation of the house. Just think about the basement membrane as the foundation of the house. So it's the foundation for the epithelial tissue itself. It's a semi-permeable band-like structure that is made up of a number of components that are largely extracellular matrix in form. So it's just extracellular matrix that tends to be found beneath the epithelial tissue. There are majorly two layers of the basement membrane. There's a lamina lucida and the lamina densa. This is a picture of it. So the basal membrane can also be called the basal lamina. So in case you see this word being used, please don't be confused. There's also another way of saying basement membrane. So look at the basement membrane here. You can see the lamina lucida, which is the part closest to the epithelial basal pole. And then we can see the lamina densa, which is the part that is closest to the connective tissue below it. And remember that between what tends to anchor the epithelial tissue, the basal part of the epithelial tissue, to the basement membrane are those cellular junctions I mentioned called hemidesmosome. This is the hemidesmosome here, connecting them to one another. The last feature of the epithelial tissue is in the ability to regenerate. I mentioned this before. Epithelial tissues tend to have a unique set of germ cells, or we call stem cells, that tends to replace it. These cells are mitotically active. They are always dividing. They're frequently undergoing cell cycle and cell division. And as a result, they tend to always replenish the epithelial cells. And this is unique because of the fact that remember that the epithelial cells tend to provide protection, mechanical protection to the surfaces that they tend to line. And this is important because as we have mechanical wear and tear, these cells are usually lost and they need to be replaced. So it's unique, it is unique and it is timely in the sense that those germ cells, stem cells are present there to just replace epithelial cells that have undergone wear and tear due to its ability to protect surfaces in which it lines. These regenerative cells, the germ cells, tend to lie very superior to the basal lamina. So let's say we have layers of epithelial cells lining a surface. Let's say these are the layers of epithelial cells. Let's use the skin as a typical example. The skin has about 5 layers of epithelial cells that lines it. The lowest layer of cell lining the lowest layer of epithelial cells lining the skin is the one that is closest to the basal lamina or basement membrane. Let's imagine this is the basement membrane here. The layers of epithelial cells found here are the ones that are going to give rise to the stem cells, are the ones that are forming those regenerative germ cells that tend to replenish the entire epithelium of the skin. So if we talk about the skin as a topic separately, we'll make mention of this again. That layer is usually known as the stratum basalis. So stratum basalis is the um, lowest layer, is the layer just above the basal lamina and is involved in the production of stem cells and replenishing of the entire epithelial tissue of the skin. Let's talk about the types of epithelial tissues we have. We have two major types of epithelium, and this is based on function. We have surface epithelium that tends to serve the function of lining surfaces, whether outer surfaces or inner or internal surfaces. And then we have glandular epithelium that tends to serve the function of creating the structural architecture of a gland. Let's start with the surface epithelium. Like I said, these are epithelial tissues that line surfaces, they line organs, they line blood vessels, they line the skin. They also line body cavities, like I mentioned before, the pleural space, the peritoneum, and the pericardium are all lined by this surface epithelium. How do we classify surface epithelium? There are two major ways we can classify surface epithelium. We can classify based on the thickness, or we can classify them based on the shape. When we classify the surface epithelium based on thickness, we have two major forms. We have the simple surface epithelium. This is just tissue or epithelial tissues made up of just a single layer of cells. So if I to draw that, for example, this is practically how simple surface epithelium will look like. Just a single layer of cell. So we have a number of cells. You can have about 10 to 20 cells here making up the tissue. Each of them are just on the same horizontal plane or same layer. Then we have the stratified epithelium, stratified epithelium is when we have more than one layer of cell, like what I talked about for the skin. The skin is an example of the stratified epithelium because in the skin we have multiple layers of cells that are even grouped. So we have majorly five um, major layers of cells found within the skin. So let's use that as a typical example of stratified. Since we're having five, each of them have different names. We have the stratum corneum at the top. This is made up of a number of cells that are they are even more than just a single layer. So we have multiple layers of cells forming the stratum corneum. Then we have the stratum lucidium, we have the stratum granulosum, we have the stratum spinosum, and then I mentioned that the last one, which is the stratum bisalis, which is the layer that is involved in germ regeneration. So this is a typical example of a stratified epithelium because we have multiple layers of cells horizontally arranged that tends to form the entire epithelial tissue. We can also classify epithelium based on surface epithelium based on shape of the cells. So some cells or some epithelium tend to have the cells being squamous in shape. The squamous shape of the epithelium tends to be a flat type of cell that is wider than taller and is used. Usually, the cells tend to be used for the process of diffusion, for the process of cell permeability. They are the cells that allow substances to go to and fro uh, within a substance in which they are lining. A typical example with squamous epithelium would be the lining of the lungs. The lungs tend to have squamous epithelium lining to alveoli, and it allows for um, movement of oxygen and carbon dioxide across the entire lung tissue. Then we have the cuboidal cells. Cuboidal epithelium are epithelium that have cells that are cuboidal in shape. They are taller than they are taller as they are wider, so they are almost as tall as they are wide. So this is contrast to squamous, in which they are wider but shorter, so wider than they are taller. For cuboidal, they are tall as they are wide, so they are almost the same width and height. And usually, cuboidal epithelium tend to have the function of secretions. They are involved in the process of secretion. Most of the cuboidal cells that we see or cuboidal forms of epithelium that we see tend to also be similar in architecture to the glandular epithelium because of the fact they are involved in secretions. And then finally, we have the columnar epithelium. Columnar epithelium contains cells that are columnar in shape. They are taller than they are wider, so they have a larger length compared to their width. And these cells are involved in absorption. So that's the cells that we see lining the epithelium of the intestine, which is involved in absorption of nutrients into the bloodstream. So these are just examples of each of the different forms of epithelium. I've divided them based on those two major classification criteria into whether they are simple or stratified, and also based on the shape, whether they are squamous, cuboidal, or columnar. Look at some examples of each of the different organs that are lined by these forms of epithelium. So we have the simple squamous epithelium found in the alveoli of the lungs, like I mentioned, and the glomerulus of the kidney. These two are important for diffusion. Remember that lungs is involved in diffusion of oxygen and carbon dioxide. The glomerulus is involved in filtration of substances within the blood, and that entails diffusion as well. For simple cuboidal cells, this one is simple cuboidal cells is involved in is found in the tubules of the kidneys, so proximal tubule and distal tube of the kidney, and is also found in the genital tract of males. Simple columnar epithelium is found in the stomach and intestine. I already mentioned the fact that they're involved in diffusion, in absorption rather. So for them to be able to be undergo absorption, they need that simple columnar epithelium. That configuration tends to increase the ability of the organ to absorb different substances. Then stratified squamous epithelium. Examples of sites where we have stratified squamous epithelium could be the skin, could be the tongue, the gums, the vagina, and the esophagus. And usually we tend to distinguish or sub-distinguish stratified squamous epithelium into keratinized version, which are the ones that have keratin. Keratin are just is just a protein substance produced by the epithelium to make it a bit tougher. So areas that tend to have a lot of mechanical wear and tear, like the skin, the tongue, the gums, tend to have keratinized stratified squamous epithelium. Whereas areas that tend to have very little wear and tear, mechanical wear and tear, would have non-keratinized stratified squamous epithelium, like the vagina and the esophagus. Then we have stratified columnar epithelium. Stratified columnar epithelium are found in areas like the trachea, the bronchus, and the cervix. And then finally, transitional epithelium. I didn't mention this before, but this is just a unique form of epithelium in which we have a combination of the columnar squamous epithelium and cuboidal epithelium all arranged in a fashion in which all of them are in one piece, they're in one tissue. So it's like there's a transition from columnar to squamous as we go from down to up. This is very unique to the urogenital system, to the urinary system to be specific. So urinary system entails the bladder, ureter, and the urethra. Each of these organs contain transitional epithelium. Let's talk about the glandular type of epithelium. These are single cells or groups of cells that are forming tissues that tends to produce and secrete specific biological substances, which we call glandular secretions. We have two major types of glandular epithelium. We have the endocrine glandular epithelium, which are found in endocrine glands, and we have the exocrine glandular epithelium found in exocrine glands. So just to distinguish each of the different forms of glandular epithelium, starting with the exocrine gland. Exocrine glands usually tend to be ductless glands. They're glands that lack a duct. They tend to secrete their secretions into the bloodstream, and the secretions are usually known as hormones. And examples of endocrine glands include the thyroid gland that produces thyroid hormone, the adrenal gland that produces cortisol, aldosterone, and some androgens, and then the pituitary gland that is involved in producing a number of different hormones. And then we have the exocrine glands. Exocrine glands are usually glands with ducts. They are glands that tend to empty their epithelial secretions into those ducts, and those ducts will eventually um, drain the secretion into an epithelial surface. Some of the secretions that are produced by exocrine glands include that of sweat produced by our sweat glands in the skin, sebum, oil produced by the sebaceous gland also in the skin, saliva produced by salivary glands, and some other glands like the pancreatic secretion produced by the pancreas, and some intestinal secretions as well. So examples that already, like I mentioned, the salivary glands are examples of exocrine glands producing saliva. Mammary glands are glands of the breast that are involved in producing breast milk. So just as we classified the surface epithelium, let's classify some of the glandular epithelium, but we will only focus on the classification of exocrine glands because these are the only ones we can classify. Usually, endocrine glands tend to occur in clusters, and we don't really have a unique form of classification for each of them. So we will talk about classification of glandular epithelium by focusing on the exocrine glands. And there are five major ways we can classify an exocrine gland. We can classify it based on the branching, we can classify it based on the shape, we can classify based on type of secretion, we can classify based on the arrangement of the cells, and we can classify based on the mode of glandular secretion. So based on the branching of an exocrine gland, glands could either be classified as a simple or compound gland. A simple exocrine gland is one that is unbranched, has unbranched ducts. This is a single or multiple glands with unbranched duct or singular duct. And this is a picture of how that looks like. You can see each of these glands are singular ducts that are draining it, and they are unbranched. Whereas a compound extract gland is one that has multiple ducts, could have multiple ducts, and each of those ducts have branches. You can see, although we have a single duct at the end of this portion, but you can see each of these ducts are branched, and as a result, we have multiple ducts that are branched together to form the compound nature of the extract gland. Based on shape, we could have the glands being tubular, alveoli, or tubulo-alveoli. Tubular, just as the name implies, it looks like a tube. As you can see, this is an example of a tubular gland. Looks exactly like a test tube. Alveolar glands are glands that looks like like a bunch of flax or grape. So if you imagine a flax would have like a long end and then a circular bottom. The circular bottom tends to be the aspect that tends to have that alveolar sac or flax-like shape. So the alveolar glands tends to be unique in comparison with the tubular gland. And then we have the tubulo-alveolar gland, which is a combination of both of them. So this gland would have both tubular and circular architecture of its glands. So based on type of secretion, we could have a gland, an exocrine gland being a serous gland. So it's involved in producing secretions that are largely watery and contains large amount of enzymes. So an example is a salivary gland that has a number of enzymes and is very watery. We could also have mucous glands. Mucous glands are glands that produce a lot of mucin-rich secretions. So these are fluid-like secretions that are not as watery as the first gland, but contain a lot of mucin. Mucin is a glycoprotein that tends to be combined with water. And then we have a mixture. We have a mixture, a mixed cells and mucous gland. Based on the arrangement of cells, we have cells being unicellular or multicellular in nature. So examples of glands that are unicellular could be goblet cells. Goblet cells are found within the small intestine. They are examples of glandular epithelium, but are just a single cell. So they are single cell gland that helps in production of mucous gland or mucous secretions. Then we have multicellular glands. So examples of multicellular glands are some of the ones we talked about, like the tubular glands, alveolar glands, and the mixed glands. So glands like the Bronner's gland, the duodenum, is an example of a tubular multicellular gland. Glands like the sebaceous gland, in the skin, are examples of alveolar multicellular gland. So glands like the salivary gland are examples of tubular-alveolar multicellular gland. Finally, the mode of secretion. So based on mode of secretion, we have exocrine glands being classified as either an apocrine, merocrine, or holocrine gland. And this tends to be very important to remember. For merocrine glands, they tend to have secretory products that are produced by way of vesicular exocytosis. So for merocrine glands, it's through exocytosis that they tend to produce this secretion. Popular example of a merocrine gland would be that of the pancreatic cells, the sweat glands, and the saliva. For apocrine glands, the secretions are made possible by way of cytoplasmic secretions by fusion, facilitated transport, and this usually tends to be found in glands like the mammary glands, and glands within the armpits, like the axillary glands. Like the glands within the armpits are examples of apocrine glands. The glands in the breast are also examples of apocrine glands. So they have cytoplasm, and the secretions are usually tends to be excretions, not necessarily even secretions. Then finally, we have holocrine glands. Holocrine glands um, tend to have the entire cell being lost when secretion occurs. So examples of holocrine glands are sebaceous glands, stem cells that they usually have stem cells that tend to divide and replace the lost cells. So a sebaceous gland, for example, if it's going to secrete anything, it's going to lose itself in doing so. So in secreting something, the entire cell is being lost, but eventually it's replenished by a stem cell. And that's the difference between holocrine gland and the rest. And that concludes our module on epithelial tissue.